Let's start off with the primary structure. Okay, primary structure, what does that tell us? It's the sequence of amino acids. It's the sequence of amino acids. Okay, for the secondary structure, well, what's the secondary structure? The secondary structure is basically primary structure is folded, sequence of amino acids are folded into alpha helix or beta pleated sheet. And also state what bond is present. So we know that hydrogen bonding is present. Okay, tertiary structure, what happens? We've got ionic and hydrogen bonds present. We're forming a unique 3D shape. So unique 3D shape. And we've got hydrogen plus ionic bonds present between the R groups. Okay, and then the quaternary structure, what's going to happen? A quaternary structure contains more than one polypeptide chain. So a quaternary structure contains more than one polypeptide chain. Okay, so we've said 4 points there. So we're going to get 4 marks because they're nice lengthy points as well. Let's think of it now. Let's add a bit more detail. Describe how the structure of a protein, so we're talking about the structure of a protein, depends on the amino acid it contains. Okay, so the amino acids, how does the structure of a protein depend on the amino acids it contains? So we know that it depends on the sequence of amino acids and the bonds which are present, but what else? Okay, so it's basically the interactions between these bonds. Okay, have we stated interactions anywhere? Okay, we've not stated it. I'm going to state it at the top over here. It's basically, it depends on the amino acids it contains because the amino acids are positioned specifically, and there are specific interactions between the relative amino acids. That's what I'm going to put. There's going to be specific interactions between relative amino acids. Okay, if we have a look at the mark scheme over here, it says structure is determined by relative position of amino acids or interactions. Okay, That's what we put. We put interactions. I mean, we've stated these bonds, so I'm sure we would have still got the marks anyway. Primary structure, the sequence of amino acid. Secondary structure, hydrogen bonding. Tertiary structure formed by interactions between all groups. Creates active site in enzymes. Now, it's not basically stated that point. We don't know if we're talking about enzymes. That's why it's put that as an additional point, I guess. Six, quaternary structure contains more than one polypeptide chain. Okay, so we would have got all 5 marks. It's got additional points over here because I don't think many people would have picked up on this point over here, because I wouldn't have known that they were talking about enzymes within this question. Okay, next question. Okay, so we know that a dipeptide consists of 2 amino acids joined by a peptide bond. Dipeptides may differ in the type of amino acids they contain. Describe 2 other ways in which all dipeptides are similar and one way in which they might differ. Okay, so we know that dipeptides differ in the type of amino acids that they contain. What are the similarities between dipeptides? Well, basically, what do we know about amino acids? We know that amino acids all have an amino group. We all know that they have a carboxyl group. We know the structure of them. Well, and it has 2 similarities. So what's going to be the similarity between all dipeptides? Well, all of them are going to contain an amino group. So all are going to contain NH2, and all of them are going to contain COOH. So all are going to contain COOH. So what's the difference? What makes dipeptides unique? Okay, what makes an amino acid unique first? Okay, we know that everything's the same in an amino acid apart from one thing, and that is the R group. The difference is that they're going to have different R groups are going to be present, and that's the difference there. Okay, so amine or amino, carboxyl, you could have said 2 R groups, all contain CH, N, and O, and there's going to be variable groups as the difference. Okay, so think about it. Whenever they're asking you to compare dipeptides, always remember dipeptides are formed from 2 amino acids. So you're basically having to compare the structure of 2 amino acids, the similarities, and the difference. Okay, that's why I've come to this. Both of them contain NH2. Both of them contain COOH at either end. If we look at that diagram that we saw earlier on, we know that both of them contain those groups. The only difference is between the structure, and that's where the R group is, because everything else is the exact same. Okay. Next question, 2 marks. Describe how a peptide bond is formed between 2 amino acids to form a dipeptide. So again, this is basically a recall question. However, it's really careful. You have to be really careful to write in the way the mark scheme wants you to write. Okay, so how is a peptide bond formed? Well, we know that any chemical bond is formed by the condensation reaction. So condensation reaction, where does the condensation reaction occur? It occurs between the amine group of one amino acid and the carboxyl group of another. Okay, so between the amino group of one amino acid and the carboxyl group of another. Okay, so condensation reaction or loss of water between amine, NH2, and carboxyl. That would have got you 2 marks. Okay, next question. This is our penultimate question. It says the secondary structure of a polypeptide is produced by bonds between amino acid. Describe how. Okay, first of all, what bonds are present? If we're talking about the secondary structure, what bonds are present? Okay, there's only one bond which is present, and that is hydrogen bonds. Okay, so hydrogen bonds are present. And okay, so hydrogen bonds are present. Where are they present? They are present between the where are they present? They are present between the NH of one amino acid and the COO of another amino acid. Okay, so they're present between the NH of one amino acid and CO of another amino acid. And what's actually happening, the hydrogen bond is forming from the H to the O. Okay, that's where the hydrogen bond is actually forming from. Okay, it's forming between those two groups, but specifically the hydrogen is bonding to the oxygen from the COO. Okay, looking at the Morse scheme, so hydrogen bonds are present, as we know, between the NH group of one amino acid and the C double bond O group of another. You could have also said forming a beta pleated sheet or alpha helix. Reason why is because this is the secondary structure, and it says describe how the secondary structure is formed. And the secondary structure is basically the alpha helix or beta pleated sheet. Okay, final question, guys. Two proteins have the same number and type of amino acids, but different tertiary structures. Explain why. Okay, now this brings back the example that I put in forward. Remember when we had the square, the triangle, the circle, the square, the triangle, and the circle. And then we had the triangle, the square, the circle, the triangle, the square, the circle. What's the difference about this? We have the same number of amino acids, we have the same type of amino acids, but we have different tertiary structures. The reason why is because we have a different sequence of amino acids. Okay, so we have a different sequence of amino acids. Because we have a different sequence of amino acids, what's going to happen? If we have a different primary sequence, primary structure, we're going to form our ionic and hydrogen bonds in different places. So the ionic, always name the bonds. The ionic or hydrogen bonds are going to form in different places. Okay, looking at the morpheme, different sequence of amino acids or different primary structure forms ionic hydrogen or disulfide bonds in different places. Okay,